Just nice to see you. Today we will see about the membrane and cell physiology. So this is a lecture outline. This is the objective. So we have seen chapter 1, Introduction to Physiology. So today we will see about the membrane cell physiology, and next we will see about the membrane physiology. So it's very an important section. So please be with us. Cell in general, the structure of cell seems like just like this. So now we do have two types of cells. The one is the cells without typical nucleus. The other is with typical nucleus. Cells also have true nucleus. We can call it eukaryotes. Eu means true, karyotes means nucleus, true nucleus. This is pro means before. So prokaryotes means there is no typical nucleus, but in the case of eukaryotes there is true nucleus. So we will have when we compare and contrast the eukaryotes and prokaryotes, there is nucleus in the case of eukaryotes, but there is no nucleus in the case of prokaryotes, and their size is eukaryotes are larger than prokaryotes, because the eukaryotes have 10 up to 100 micrometer in size, and there is cytoskeleton in eukaryotes and no cytoskeleton in prokaryotes. Membrane bounded organelles are present or found on eukaryote cells, and no membrane bounded organelles are found in prokaryotes. And RNA synthesis is in the nucleus, protein synthesis is in the cytoplasm in the case of eukaryotes, whereas RNA and protein synthesis occur in the cytoplasm in the case of prokaryotes. So multiple linear or chromosomes are found in the case of eukaryotes, whereas smaller circular chromosome is found in the case of prokaryotes. So this is cell. In general, cells are we do have three types of three segments or three parts of cells. The one is cell membrane, the other is here cytoplasm, and the other is here nucleus. We do have three parts. So the boundary between the nucleus here, the nucleus and cytoplasm is called nuclear membrane. Here is in the nuclear membrane, and also here is cytoplasm, and cytoplasm is bounded or separated from the extracellular fluid by cell membrane, or another name of cell membrane is plasma membrane. So the overall cells are made up of different types of substances. This collective name is known as protoplasm. So protoplasm is composed of 5 basic substances. So these substances are water, electrolyte, and protein, lipid, and carbohydrates are considered as contents of cells or protoplasm. So having this concept, cells are the structural and functional units of the living thing. So cells are the smallest living thing, and fields which study about cells are cytology, and cells are perform all activities necessary for survival, growth, and reproduction of organisms. So in cytoplasm there is cytosol, that fluid compartment is called cytosol, and cytoskeleton, that is fibril like microtubules, intermediate filaments, and microtubules, intermediate filaments, and other types of tubules are considered as cytoskeleton, fibril components. The others are here, all are organelles and also inclusions. Organelles are living materials, whereas there are also inclusions. These inclusions are lifeless materials like protein, carbohydrates. and this covering is plasma membrane. So let me show you about the organelles which is found on the cells. This is nucleus. This is known as nuclear membrane, and here is nucleolus. Here is nucleolus, and here is chromatin. These teeny dot-like particles are known as ribosome, and here is known as rough endoplasmic reticulum, because of these teeny reed-like dot-like structures are known as ribosomes. So endoplasmic reticulum, those who ribosomes attached to it is called rough endoplasmic reticulum, whereas there is no ribosome here. This is known as smooth endoplasmic reticulum. And whereas here is mitochondria, here is lysosome, and here is cytosol, and here is tube-like structures are centroles, and this is microtubule, and this is intermediate filamentous, and this is periosome, and here is these structures are known as Golgi apparatus, and here is ribosomes, and this is rough endoplasmic reticulum, and this is nuclear envelope, and all are this is we can say organelles of the cell. So now this is a cell membrane which differentiates cytoplasm from extracellular fluid or intracellular fluid from extracellular fluid. So this is about 7.5 millimeter thick and flexible barrier that surrounds the cell, and it's composed of protein and lipid bilayer. So it is semi-permeable membrane. This is overall about the cell structure, and the first thing is here we will see this one, plasma membrane or cell membrane. So plasma membrane is a sheet-like structure that surrounds the cell, and it separates cellular contents from extracellular matrix. That means it separates from intracellular fluid from extracellular fluid, or separates cytoplasm from extracellular fluid, and regulates the passage of substance in and out. It serves as a gatekeeper that regulates the passage of substances in and out of the cell, and also the other functions of cell membrane is also used as a sensory device that permits the cell to recognize and to be recognized and by other cells, and also used for cell-to-cell attachment and communication. And plasma membrane is also divided and here, later we will see later. So here is nucleus. Nucleus is the central part that we have seen. It's a control center for cells. And it contains genes, which are units of heredity. That means the genes are important to transmit its heredity from one generation to the other. Chemically, each gene consists of highly composed of DNA in the form of chromosomes. So genes control cellular activity by determining the type of protein, enzyme, and other substances that are made by the cell. So nucleoli is a site for ribosomal RNA synthesized. So nuclear contents are surrounded by double-wall nuclear membrane, as I showed you before. The pores present in this membrane allow fluids, electrolytes, RNA, and other materials to move between nuclear and cytoplasmic compartments. So nucleus with DNA coordinates protein synthesis in ribosomes, and transcription phase of protein synthesis occur there. So DNA is changed into RNA by process of transcription, and also RNA changed into protein by process of translation in the ribosome. This is the nucleus. Within the nucleus, this is in the ribosome. So following transcription, messenger RNA leaves the nucleus and travels to the cell's ribosome, where translation occurs. So transcription is occur within the nucleus, whereas translation occurs in the ribosome. And cellular organelles. As I told you before, cellular organelles are found within the cytoplasm. So organelles are ribosomes, endoplasmic reticulum, Golgi apparatus, mitochondria, lysosome, and cytoskeleton system. So and from this ribosomes, microtubules, and centrolus filaments are cilia and flagella are non-membranous organelles, whereas cell membrane here and mitochondria, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, secretory vesicle, lysosome, coated vesicle, endosomes, periosomes are considered as membranous organelles, which is covered by membrane. Cytoskeletons are composed of three. The one is microtubule, microfilament, and the intermediate filament. So they told you before, here all organelles are mentioned here. So now another option is this picture. So this is cell membrane, this is a nucleus, this is nucleolus, and this is rough endoplasmic reticulum, and this is smooth endoplasmic reticulum. This is lysosome, this is mitochondria, whereas this is microfilament, this is centrosome, and this is microtubule, this plasma membrane, this is mitochondria, and this is ribosomes, and this is chromatin, and this is Golgi apparatus, Golgi vesicle, and cytoplasm, vacuole, periodicosome, secretory vesicle. This is organelles, which is found between the nucleus and the membrane, cell membrane, and this is structure of human cell. So now we are when we are observing organelles, so ribosomes, the sites of protein synthesis in the cell, so found into forms. The one is attached to the wall of endoplasmic reticulum, the other is as ribosomes, as free ribosomes. So we can divide into ribosome into two. The one is attached ribosome, the other is also free ribosome. So attached ribosome means that ribosomes that have a connection or association or attachment with endoplasmic reticulum. These attached ribosomes are attached to endoplasmic reticulum. That's why we call it rough endoplasmic reticulum. So, and some of are an outer nuclear membrane, attached to outer nuclear membrane. So this is involved in the synthesis of protein designed for insertion in the plasma membrane or for export from the cell. So attached ribosome is important for plasma membrane, for structure, keeps structure of the plasma membrane or exported outside the cell. Whereas these free ribosomes are float in the cytosome and have no attachment to other organelles, and its function is to synthesize protein for use inside the cell. So free ribosome is for inside the cell, attached ribosome is outside the cell or for cell membrane. So for cell membrane, the site of it is the site of protein synthesis. That means messenger RNA is translated into protein. So free ribosomes either found in scattered form or found in clustered or aggregated form. So ribosomes are here. These thin like dot-like structures are ribosomes here. So this is free ribosome, this is attached ribosomes, and the other is endoplasmic reticulum. It's an extensive membrane structure that connects various parts of the inner cell, and endoplasmic reticulum is also connected with nuclear membrane, having association with the nuclear membrane. There are two types of endoplasmic reticulum. The one is rough endoplasmic reticulum, the other is smooth endoplasmic reticulum. that rough endoplasmic reticulum is named. It gets its name in association with ribosomes, because ribosomes are attached to it. So we say the rough endoplasmic reticulum. So because we say that rough endoplasmic, or we can say agranular endoplasmic reticulum. Why? Because because of ribosome, and also its function is used for protein synthesis. So it's a site where proteins are synthesized. A site where proteins are synthesized. Whereas smooth endoplasmic reticulum it's free of ribosome. There is no connection, association with ribosome. So we say it is smooth. So it is important for synthesize fatty acids, steroid hormone like estrogen and progesterone. Important for drug detoxification, like in hepatocytes. That's drug metabolism or detoxification in hepatocyte cells. The other important for important synthesis of fatty acid or lipid biosynthesis. Important for calcium storage, glycogen storage, are function of smooth endoplasmic reticulum. So the other is Golgi Complex. Golgi Complex or Golgi apparatus. It is acts as receptacle or container for hormones and other substances that endoplasmic reticulum produces, and it modifies, packages these substance proteins into secretory granules. So the main functions of Golgi complex is processing of proteins synthesized in the rough endoplasmic reticulum, and it is important for retrieval, recycling, membrane retrieval, recycling, and redistribution. So the other important organelle is mitochondria. Mitochondria are literally the power plant's factory, or we can say that the powerhouse of the cell. And because many reactions that produce energy take place in the mitochondria, take place in the mitochondria. The major site of mitochondria is the major sites of ATP production and oxygen utilization. So ATP is an energy producing, an energy part of the cell that important for many cellular activities. So this is an energy for many activities of the cell. Continuous enzymes of Krebs cycle and oxidative phosphorylation. Contains enzymes of Krebs cycle and oxidative phosphorylation. So ATP, 2 ATP, 36 ATP are found when it's produced through the Krebs cycle. So the mitochondria is, by the way, it's a very important organelle. So the one is it's important for generate ATP. That is, we say it powerhouse of the cell. The second is important for heat production in the brown fat cell. And also there is a unique properties of mitochondria is possess their own genetic apparatus and proliferate by division or fission of pre-existing mitochondria. This is a function of mitochondria. And the other organelle is known as lysosomes. Lysosomes are very important organelles. Lysosomes are vesicular organelles that form by breaking from Golgi apparatus, then dispersing throughout the cytoplasm. Contains aggregates of enzymes. Lysosome contains more than contains more than 40 powerful enzymes, hydrolyses, which are synthesized in the rough endoplasmic reticulum. So aggregate enzymes is having more than 40 powerful enzymes. These enzymes break down wide variety of molecules and like bacteria substances. So will develop in macrophages. They are called suicide bugs. they provide an intracellular digestive system that allows the cell to digest and damage cellular structures, harmful substances such as bacteria. So it's known as suicide bugs. And the other is lysosomes are when compared with the peroxisomes. Compared with peroxisomes, lysosomes comes from Golgi apparatus. And lysosomes are membrane-bounded organelles that contain hydrolysis or hydrolytic enzymes. Hydrolytic enzymes are like lipases, proteases, and glycosidases and nucleases are considered as lysosomes or hydrolytic enzymes. So the membrane surrounding them prevents enclosed hydrolytic enzymes from coming in contact with other substances in the cell and therefore prevents their digestive actions. The other organelle is known as peroxisomes, or you can say small bodies. It is a single membrane bounded organelles containing enzymes, oxidases and catalysis, and secrete chemicals that converts harmful substance into harmless. For instance, ethanol is detoxified by peroxisomes. So catalase is a type of oxidase produced by peroxisomes and converts hydrogen peroxide into water and oxygen. This is harmless or useful. So hydrogen peroxide is acid, strong acid. So by catalase changed into water and oxygen. So this is the functions of peroxisome. So peroxisomes are similar physically to lysosomes, but they are different in two ways. They are formed by self-replication, perhaps bounded off from smooth endoplasmic reticulum. So it is formed by self-replication and rather than from Golgi apparatus. So arise from Golgi apparatus is for ribosome, and smooth endoplasmic reticulum is for peroxisome. They contain oxidase rather than hydrolase, so oxidase enzyme, whereas lysosomes are hydrolytic or hydrolase enzymes. Hydrolytic enzymes. So this is the main difference between the two organelles, peroxisome and lysosomes. So other type of organelles are membrane we will see later. The other is centrols and inclusions, life materials. Cytoskeletons are microtubules, microfilament and intermediate filaments. They are important for maintaining cell shape, stabilizing cell attachments, and promote cell motility. And the other is also nucleus. I told you nucleuses are very important. They have nuclear envelope, nucleolus, nucleoplasm, chromatin. Within chromatin there is DNA and RNA, and so nucleolus also contain DNA and RNA. Now we will go into so we have we have completed about cell organelles. So the other we will see about cell-to-cell communication, how one cell is communicate with other cells. Cell-to-cell communication is very important for multicellular organisms. So the communication is through endocrine signals, paracrine signals, and autocrine signals. And the other signal is we do have also 3 types of signal. Also there are other types of signals. This is known as juxtacrine signals. Juxtacrine signals are composition of paracrine and autocrine signals. So endocrine signals are done through hormones produced by endocrine cells. So endocrine cells are endocrine cells are found on the endocrine organs like pituitary gland, thyroid gland, parathyroid gland, pineal gland, and adrenal gland. And hormones travel through blood to reach their target cells. So hormones, for instance, hormones that's produced by pituitary gland is goes into blood stream, so mixed with blood and transported through the body until it reaches into its target cells. So the other is paracrine signals. This target only cells in the surrounding area of releasing cell. That is paracrine cells are surrounding area of releasing cell. So an example is neurotransmitters. And the other is autocrine signals, affect only cells that are of the same cell type as emitting cell. So the autocrine cells are cells which have similar the cell its own type cells. An example is immune cells. An example is immune cells. The other type is juxtacrine cells. This is contains it have characteristics of paracrine and autocrine signals. They are capable of affecting either by emitting cell or cells immediately adjacent to it. So they are transmitted along cell membranes via protein or lipid components integral to the membrane, and are capable of affecting either emitting cell or cells of immediately adjusted. So we do have almost 4 types of communications. So this is hormone secreting gland cell, secretes hormone, so go into bloodstream, and it affects the target cell. This is endocrine signals. And the other is nerves. This is nerve impulses, neurotransmitter, then affects it is neuron or effector cell. And this is known as paracrine signals. So neuro hormone, a target cell, and local cell, paracrine agent. This is target cell. So this is paracrine signals, and this is autocrine signals. It affects cells similar to it. And this is cells affected by target only cell surrounding area of releasing cell. So these two characters also known as juxtacrine cells. The other types of section is junctions between cells. How junctions between cells are formed and how many junctions are present. So multicellular organisms require specific interaction between cells to hold the cells together, to attach or hold cells together, and also to communicate in order to coordinate activities. So this is done through junctions. We do have almost different types of junctions, almost 5 kinds of junctions, tight junctions, gap junctions, desmosomes, and hemidesmosome, and adherent junctions. But commonly known is the tight junction, gap junction, and desmosomes. Let me show you an example of these junctions. So, by the way, this is a plasma membrane, double plasma membrane. So extracellular space is here. This is extracellular space or outside space. This is the membrane of 2 cells. So this is known as tight junction. This is tight junction. The 2 membranes are already overlapped. So this is a tight junction. In the case of tight junction, there is no plasma membrane adjacent cells to fuse together, so that it is impermeable. So there is no material to pass. So passing through extracellular space between adjacent cells are prevented. So maybe molecules are passed either through diffusion or active transport. So tight means it's impermeable. So this is tight junction. And the other is desmosomes. Desmosomes are, this is membrane, this is open. So desmosomes are here. So this is Lysosomes here, and this is mostly found in the skin, higher and pure in present. So this is a gap junction. This is one, so this is a gap junction. This is gap junction. So gap junction is a communicating junction between adjacent cells, this cell 1 and cell 2. So this is a gap junction. This is very important gap. Allow ions and molecules to pass for intracellular communication. So this is found on the smooth muscle and cardiac muscle. This is known as this channel. This tube is known as connect zones. So this is a hollow cylinder that connects to membranes. So this is very important. It's another name is known as communicating junction, because it allows ions and molecules to pass for intercellular communication. So this is cell 1 membrane, this is another cell membrane, cell 2 membrane. So when we are good to the other, in theory this is tight junction or occluding junction. Tight junctions help plasma membrane of adjacent cells to fuse together, forming impermeable junction. Impermeable means it's not permeable to pass anything, to pass ions or molecules from one part to the other, and prevents molecules from passing through extracellular space between adjacent cells, between two cells. As it is, most materials must actually enter the cells either by diffusion or active transport. So this type of junction prevents toxins from leaking into the bloodstream. So an example is tight junctions between epithelial cells. So tight junction, an area where tight junction found is between two epithelial cells. So it is found in epithelial cells between two adjacent epithelial cells. It is a common. The other is a gap junction or communicating junction. It is a communicating junction between adjacent cells, between cell 1 and cell 2. At gap junctions, the adjacent plasma membrane are very close, and cells are connected by a hollow cylinder. It's called connect zone. Allow ions and molecules to pass for intercellular communication between two cells. Inter means between two cells. So an example where gap junction found is between smooth muscle cells and cardiac muscle cells. So it is present in electrically excitable tissues such as smooth muscle, heart, where ion passage from cell to cell helps synchronize their electrical activity and contraction. And the other is Lysosomes, mostly found in tissues that are subject to constant mechanical stress, such as skin and heart. Lysosomes are localized patches that hold two cells tightly together. So they are common in epithelium, that means especially skin. So skin is it's a place where mechanical stress is found. So Lysosomes this is about junctions. So this is all about cells in general, and now we will go to about the membrane or membrane physiology. Membrane or cell membrane. Now the next section, chapter 2, next portion of the chapter 2 is about membrane. The membrane. So membrane is composed of proteins, lipids, and carbohydrates in different proportions. So protein, lipid, and the most common membrane content is the protein that in cell that's next to water. So protein, next to protein is lipids and carbohydrates. So 55% of proportion of cell membrane is protein, and the other 42% or the second type is known as lipid, and the other 3%, the lowest one, is carbohydrate. And lipids are also 25% of lipids are, or from this 40% is phospholipid, and 30% is cholesterol, and 4% is neutral fats. Neutral fats. So the other thing is functions of plasma membrane. One of the functions of plasma membrane is separate cellular contents from extracellular fluid, separate intracellular fluid from extracellular fluid, or separate cytoplasm from extracellular fluid. Regulate the passage of substance in and out. This means it used as it is a gatekeeper that regulates the passage of substance in and out of the cell, and it is semi-permeable or selectively permeable membrane, allowing some substances to pass through it, excluding others. This creates an equal distribution of ions on both sides of the membrane. It allows oxygen and nutrients to enter the cell while keeping toxins and waste products outside the cell. So it produces receptors for neurotransmitters, hormones, and drugs. It's a means to cell-to-cell contact, or it's important for cell-to-cell attachment and communication, and for generation of and transmission of electrical impulse in nerves and muscle, as well as used as sensory device that permits cell to recognize and to be recognized by other cells and macromolecules. All these are functions of cell membrane or plasma membrane. And so we will see about the components of cell membrane. The first component is lipid, and the second is protein. So lipid and protein. So membrane lipids are composed of phospholipids, glycolipids, and cholesterol. Lipids form the basic structure of membrane. The lipid molecules are arranged into parallel rows, so it's considered as lipid bilayer. So there is protein in inner, and outside inside is formed by lipid, so lipid bilayer forms in this way. And so phospholipids, glycolipids, and cholesterol is a part of membrane lipids. And now we'll see. Now we are observing just phospholipids. Polar phosphate head. This is phospholipids, so this is known as polar phosphate or head, and the other is this is head or polar phosphate is known as hydrophilic. Hydrophilic means water loving, so living in water. So this is water loving. So this is attracted to water, so it is water loving. Whereas fatty acid or tails, this is fatty acid or tails. So this is hydrophobic. Hydrophobic means that does not mix water. That means it does not soluble in water, repelled by the water, and arranged as bilayer. I will show you how bilayer is formed. Yeah, it is bilayer. This is one layer. This is one layer. It is known as bilayer. So this is all our heads, hydrophilic heads, and these are hydrophobic tails. This is tail, this is head. So this is tail, this is head, and together this is phospholipid bilayer. This is extracellular fluid. So this is extracellular fluid, and also this is intra, and this is intracellular fluid, and this is extracellular fluid. This is between two layer. And having this concept, the physical orientation of lipid bilayer structure is that hydrophilic ends of the lipid bilayer molecule line up facing intracellular fluid and extracellular fluid. As I told you, this is head, head, this is extracellular fluid, this is head, this is also intracellular fluid. So they are lining intra and extracellular fluids heads, whereas hydrophobic tails of molecules face each other in the interior of the bilayer. So this is face each other in interior and internal part of the layer. So tails are face each other, whereas these are heads are facing extracellular and intracellular fluids. So this is aqueous, this is intracellular, this is extracellular, this is heads, polar heads, whereas this is tails. Tails are face each other, heads are towards intracellular fluid because of its water loving. But this one is hydrocarbon tails. Hydrocarbon tails. Now it's arranged as phospholipid bilayer. So this is extracellular fluid. So this is extracellular and this is intracellular fluid, and this is tails, and this are tails. So this is polar hydrophilic heads that loves water, and this is a polar hydrophilic heads that loves water, whereas this non-polar hydrophobic tails. So this cell membrane are used as cellular barrier or border. So different, this is in extracellular fluid there is sugar, water and salt here, and waste material and other waste material and others are found in intracellular and extracellular fluid. So this is impermeable to polar molecules, the tail one. So permeability to polar molecules, membrane became semi-permeable via protein channels. So semi-permeable means selectively permeable. So membrane became semi-permeable via protein channels. Specific channels allow specific materials to pass through cell membrane. So that means each content or each material having its own specific channel. So for instance, sugar passing by this channel, amino acid passing by this channel, and water passing by this channel, and salt passing by this channel, and NH3 passing by this channel. So specific channel means having, for instance, this is inside the cell. So this is extracellular fluid outside the cell. This is intracellular fluid. So this is outside the cell. So extracellular material are sugar, amino acid, water pass through this specific channel. So this is intracellular fluids like salt and NH3. So each things are passing through its own channel, specific channel. So lipid bilayer portion of the cell membrane is impermeable to water and water soluble substance. Lipid bilayer is impermeable to water as well as water soluble substance such as ions, glucose, urea and others. On the other hand, fat soluble substance are oxygen, carbon dioxide, alcohol and drugs that can penetrate this portion of the membrane. So the whole, this is our cell membrane. So this is lipid bilayer, head and tail. This is peripheral protein, and this is intracellular fluid, and this is extracellular fluid, and this is integral protein, and this is carbohydrate, glycogen. This is carbohydrate that's attached to the integral protein, and this is integral protein in general. So this is lipid bilayer. So this is impermeable for ions, glucose and urea and others, and also they are permeable for these contents like oxygen, carbon dioxide and alcohol. And before that, cholesterol is a part of lipid bilayer, so it's important for to stabilize the restructure of the cell. So that stabilizes the cell membrane and reduces neutral mobility of the complex lipids in the plane of membrane. So determine rigidity of the membrane used to stabilize cell membrane. That is cholesterol. And now protein membrane proteins or proteins. This is about the protein. We can divide into proteins into two: intrinsic or integral protein, and peripheral or extrinsic proteins. We can divide into two. So for instance, when we see its specific function, integral proteins are important to form channels, pores, so which certain substances flow into it and outside the cell. Some act as transporters, carriers, to move substance from one side of the membrane to the other, and the some serves as recognition site, as receptors. And peripheral proteins do not extend across cell membrane or phospholipid bilayer located on the cytoplasm aspect of an early fluid. Function are peripheral proteins functional. Some are enzymes, some are also cytoskeleton anchors, attach plasma membrane filaments, and intracellular messenger system. So intracellular messenger system, cytoskeleton anchors, and some are enzymes. So when we are go to in detail, integral proteins exist as globular units running through the width of the cell membrane. The width of totally run with the width of cell membrane. So it is used as channel. So partly it is hydrophobic, and partly it is hydrophilic, and partly it is hydrophobic. So hydrophilic means polar and protein to cell surface, whereas partly hydrophobic, it's non-polar and embedded in the liquid by layer. So that means integral protein have hydrophobic and hydrophobic parts, and protruding part may often carry carbohydrate chain or lipids attached to their tips like flags. So that means either carbohydrate or lipids are attached to the protruding parts of integral protein, tightly associated with membrane, and account for about 70% of membrane protein. So about 70% is 70% of membrane protein is integral, whereas 30% of membrane protein is peripheral protein. So when we see the peripheral protein, it is hydrophilic and readily dissociated from the membrane, whereas integrated is associated with the membrane. And peripheral proteins are free, floating on the surface, or stored inside and outside the membrane. That means it does not extend fully into the membrane, does not cross the membrane, either found on the outside the membrane or inside the membrane. So it's account 30% of the membrane proteins. Bind specific hormones and proteins on the cell membrane. So peripheral proteins that bind to intracellular surface constitute to cytoskeleton. So peripheral or extrinsic proteins are just like this. So when you see this is membrane, so this is a lipid bilayer, this head. So this is lipid bilayer, whereas this is integral protein. Why? Because integral, because it is fully extend across the length of the cell membrane. So this is also integral protein. So integral protein, whereas this is cholesterol. It's important to maintain cell membrane, stabilize cell membrane. So whereas this is peripheral protein, also found on this area, either found inside the cell or outside the cell. It is floating, it is free, it does not extend the whole cell, the whole membrane. So this is all about the peripheral and integral protein. And when we see the third type of constitute of membrane is called carbohydrate. Carbohydrate attached on the outer surface of the membrane, binding with protruded integral protein. If bind with the integral protein, we can say glycoproteins, whereas bind with lipid, we can say glycolipid. So the function of carbohydrate is it is a site of receptors, for neurotransmitter, hormones and drugs, cell-to-cell attachment, and important for used as receptors for recognition of one cell to the other, feel like blood groups, cells in blood groups. So it's important for receptors of neurotransmitter, hormone, drugs, and cell-to-cell attachment. So this is membrane. So this is peripheral protein. This is integral protein. So this is carbohydrate. So we can say glycoprotein. This is a peripheral protein. This is peripheral protein, and this one is integral protein because it's fully extend the whole membrane. So this is very brief example. So this is cell membrane. This is head and head. This is an intracellular fluid. This is extracellular fluid. So this is integral proteins. This is pores that ions and contents pass through outside to inside or inside to outside. And this is peripheral protein, which is found inside. And this is peripheral protein, which is found outside the cell. This is known as this is cholesterol, and this is known as glycolipid. That means lipids which attach to the this is carbohydrates which attach to the lipids, so we can say glycolipid. This is integral protein, so this is carbohydrate, so attached to the integral protein, we can say glycoprotein. This is glycolipid, meaning carbohydrate attached to the fat, we can say, or by lipid bilayer, we can say glycolipid, and carbohydrate attached to the integral protein, we can say glycoprotein. And also this is carbohydrate glycocalyx, water environment, extracellular fluid, filaments of cytoskeleton, integral proteins, and peripheral proteins. This is water environment, cytoplasm. This is extracellular fluid. This is intracellular fluid. This is cell membrane. So this is an important part of cell membrane. This is all about the membrane structure, and now we are passing through the transport through cell membrane. So the next please don't forget to subscribe this channel. So transportation across cell membrane will be seen in next video. So have a nice time. Bye bye.